Okay, let's jump right into this explainer. Look around at the incredible diversity in the biological world, from massive redwood trees towering over a forest to us just walking around, going about our daily lives. It's easy to get caught up in how different everything looks. But today, we're going to look right past all that macro-level diversity to find the underlying unity. We are taking a reductionist journey all the way down to the microscopic foundation to find the single common denominator of literally all life.
So what actually separates the living from the non-living? When you observe your environment, you inevitably bump into this giant philosophical question, like what exactly is it that an inanimate rock lacks, but a living, breathing organism possesses? Well, the answer lies entirely in life's fundamental structural and functional unit, the cell. Whether an organism is made of just one lonely cell or billions of them working together like we are, the rule is absolutely absolute. Without a complete cell, life as we know it simply doesn't happen.
And to truly understand what makes us tick, we have to look incredibly close. Think about the massive things your body does every single day. Digesting a meal, fighting off a cold, or even forming a complex memory of your childhood. Reductionist biology takes the rigid rules of physics and chemistry and applies them right to those large-scale processes. By looking at living tissues and observing the molecular mechanisms happening on a microscopic level inside a cell, we can actually decode the basis of all biological phenomena. It's like finding the source code for life.
Now, our understanding of this invisible world certainly didn't happen overnight. Back in 1838, a German botanist named Matthias Schleiden was peering at plants and noticed, hey, these are all composed of cells. Almost at the exact same time, in 1839, a zoologist named Theodor Schwann was studying animal cells. He identified what we now call the plasma membrane and also noticed that plant cells were uniquely defined by their sturdy cell walls. So together, they hypothesized that all plants and animals are composed of cells. Brilliant, right? But there was a huge missing piece to the puzzle. Where on earth did these cells come from? That mystery was finally solved in 1855 by Rudolf Virchow, who figured out that new cells are actually formed by the division of pre-existing ones.
And Virchow delivered what is essentially a true drop-the-mic moment for early biology, with this simple, incredibly powerful Latin phrase, omnis cellula e cellula. All cells arise from pre-existing cells. Thanks to that massive insight, we have the two foundational rules defining modern biology today. Number one, all living organisms are composed of cells. And number two, every single cell arises from a pre-existing cell.
To really grasp the awe-inspiring scale we're talking about here, consider mycoplasmas. They're the smallest known cells, and they are just 0.3 micrometers long. That is staggeringly tiny, especially when you contrast it with the largest isolated single cell ever recorded, an ostrich egg. It's massive. And right in the middle of those two wild extremes, you've got things like human red blood cells sitting at around 7 micrometers in diameter, and specialized nerve cells, which stretch out to be some of the longest cells in existence.
But you see, a cell's shape is just as diverse as its size, and form absolutely follows function here. Every physical shape is perfectly adapted to its specific job inside your body. Take that nerve cell. It's highly elongated to transmit electrical signals over massive distances, acting exactly like a living wire. Meanwhile, a round, biconcave red blood cell is optimized to squeeze through incredibly tight spaces to deliver oxygen. Then you have amoeboid white blood cells that dynamically change their entire shape to hunt down and engulf invading pathogens. It's purely brilliant design.
As we zoom a bit further into how these living units are organized, we can cleanly split all cellular life into two main domains. On one side, we have the prokaryotes. These are generally smaller, rapidly multiplying organisms like bacteria. They feature naked, circular DNA, meaning it's not wrapped in a protective envelope, and they lack membrane-bound internal compartments. On the flip side, we have the highly organized eukaryotes. These are defined by a distinct membrane-enclosed nucleus, extensive compartmentalization, and a whole suite of distinct complex organelles doing highly specialized tasks.
Let's look a bit closer at those prokaryotes, like bacteria. They are the ultimate biological survivors. They've got this tough, complex, three-layered cell envelope. They use mesosomes for respiration, 7DS ribosomes to synthesize proteins, and free-lying inclusion bodies just to store reserves. But honestly, the most fascinating part? Plasmids. These are extra, smaller circles of DNA separate from their main genome that act literally like downloadable cheat codes. They grant bacteria unique superpowers, like complete resistance to antibiotics. It's just an astonishing way for these simple organisms to rapidly adapt to hostile environments.
Moving over to the eukaryotic domain, the two heavyweights are, of course, plant and animal cells. While they both share that extensive compartmentalization we just talked about, they have some key differences. Plant cells are heavily fortified. They've got robust cell walls, large central storage vacuoles, and specialized equipment like chloroplasts to harvest sunlight. Animal cells, well, they completely ditch the rigid cell walls, but they distinctly possess centrioles, tiny structures that are absolutely vital for coordinating their specific method of cell division.
Okay, let's zoom in as close as we possibly can, right to the absolute outer boundary of the cell membrane. Think of it as a dynamic border control. It acts like actually, it's exactly like a microscopic bouncer protecting the internal fluid matrix. It's a selectively permeable membrane that interacts with a totally unpredictable outside world, constantly making these crucial chemical decisions about what is allowed to enter and what is strictly forced to stay out.
To put some numbers to that, let's take a human red blood cell. Its membrane is this highly tuned, complex border. It's primarily comprised of 52% proteins and 40% lipids. That leaves about 8% for carbohydrates and other helper molecules. It's a highly specific ratio that gives the cell membrane the exact structural properties it needs to survive the turbulence of your bloodstream day in and out.
And the crazy thing is, it's not a stiff wall at all. It's a dynamic fluid sea. This is the famous fluid mosaic model proposed by Singer and Nicholson back in 1972. Within this fluid sea of lipids, you have integral and peripheral proteins acting like icebergs floating laterally. They actually sail around within the membrane itself. This incredible ability to move, which we measure as fluidity, is exactly what lets the membrane do its job, enabling critical life processes like cell growth, secretion, and forming tight junctions with neighboring cells.
So, how does this microscopic bouncer actually control the border? Basically through three main pathways. Some neutral molecules effortlessly coast in along their concentration gradient, moving from high to low without the cell spending a drop of energy. That's passive transport. Water has its own special diffusion process called osmosis, just slipping right through that fluid sea. But when the cell needs to force molecules against the grain, from low to high concentration, it becomes a microscopic pump. It actively burns its own ATP energy to force those vital molecules across the border.
Which brings us to a final, pretty profound thought to leave you with today. We've just seen how reductionist biology links massive, life-defining physiological functions directly to tiny cellular mechanics. So, if your most complex human experiences, from your most emotional memories to your immune system's intricate defense mechanisms, are dictated entirely by these tiny physicochemical reactions and fluid boundaries, what other cellular mysteries are operating inside you right at this very second? It really makes you appreciate the invisible universe constantly working around the clock just to keep you alive. Thanks for joining me on this explainer. I'll catch you next time.